Hey guys, Mr. Hyatt here, and this is the second half of the Chapter 22 lecture that we weren't able to finish in class today. We left off talking about natural selection. The next step is to discuss artificial selection, which follows the same pattern, but instead of nature choosing who is fit, we choose the traits that are desirable. So we can diversify our crops and we can diversify our animals. Think about something like a racehorse. We take a big strong stud and a big strong mare, and they make big strong babies. We've got similar things with plants. We've been able to crossbreed and select out certain properties. All of these cultivars derived from this plant. So some of you maybe dislike this whole family of plants. So again, those can be attributed to artificial selection. Another example is something that we see today in pesticide and antibiotic resistance. There are going to be some of these beetles that are resistant to whatever this insecticide is. You kill off all the organisms that are vulnerable, and now you've reduced competition. These guys have plenty of food, water, shelter. They can blow up like crazy. We apply that same insecticide the following year, and it's not effective because now our population is immune. Now I'm way oversimplifying here. It's going to take more than 1 year to clear them out, and we're going to have migrants, and we're going to have mutations. We're going to have things like that's going to slow it down. But that's kind of the idea behind artificial selection. Okay, so in terms of winning in evolution or being successful in evolutionary terms, can you survive and can you make babies? Whoever lives and has kids is the winner in terms of evolution. I've talked a little bit about that in class already. So like we said, there are some underlying assumptions. We have to have variations within a population, and we have to have long periods of time. That ties back to what we talked about in class with principles of uniformitarianism and Charles Lyell and all of those sorts of things. Like we talked in class on Friday, populations evolve. Individuals can sometimes adapt. Those two things are not synonymous. Make sure you know the difference between the two. Point 2 also came up a little bit in our discussion Friday, in that only inherited characteristics can evolve. So it doesn't matter how hard I lift weights and how orange I make my skin, my son is going to come out skin color, beige, whatever color you want to call my skin, and not muscular, because those aren't inherited traits. Those aren't heritable traits. Now, we know that humans are kind of the exception to every rule. There are things like culture and learning that are acquired characteristics that sort of circumvent natural selection. Some human traits, we can afford to be obese because we don't have to chase down our food. So it would seem that obesity would make you less fit for your environment. Well, we're able to acquire food, we're able to buy food, we don't have to hunt our food down, so we sort of circumvent natural selection, and that's going to throw off our calculations if we're trying to do Hardy-Weinberg calculations. So the next section is all learning about the evidence for evolution. So these are the types of things that we look at when we're trying to figure out how evolution happens. We have direct observation, and actually these are mostly, each of these is going to get its own slide, so you can see what they're about, and we'll get some more details here in just a minute. Alright, so observations, direct observations that we see today. I've got a few details about some of these things. Let's start out looking at color patterns in guppies. Make sure you check your text for the whole write-up, and it's got some really good pictures and things like that. But essentially, when we change the selective pressure, the phenotypes that we observe change. And you can see the timeline that happened. Again, we change selective pressures, and phenotypes respond to that. Remember, evolution is just change over time. So we changed the selective pressure, the species changed, that's evolution. And there's kind of the graphical summary of that. Again, make sure you check your text for that. We also see things like HIV that mutate really quickly, quickly can develop immunity to different treatments. So if you continue the same treatment over and over and over again, it's not going to be effective, and HIV is going to continue to amplify, and it's going to continue to affect the individual. Last but not least, you guys know a little bit about the Darwin's finches. Over several years, we've taken data on beak size, and we see that in years where, let me just teach from the graph here. You see that there's a dry year. The following year, beak size decreases. Notice that every time that there's a dry year, the organisms that survive and reproduce are those that have beaks that are big enough to break open seeds. And in wet years, we see declines. So again, we change the selective pressure and the phenotype changes. We also use fossils to show evolutionary change, but the fossil record is for sure incomplete. Not all organisms are in a habitat that supports fossilization. Not all tissues can fossilize, and often these slow changes are hard to observe in what's left. And in that bottom point there, if a fossil, if that species becomes extinct, we can't track changes over time. So that sort of reinforces some of the ideas that we talked about in class. Life has changed over time. We see that some species disappear, and that sort of ties back to our catastrophism discussion. Some species failed to survive and became extinct, so the biodiversity changes over time. And I kind of talked about this stuff. Interpretation is always an issue. Two people can look at the same set of fossils and glean different information from them. Also, the missing links term is a legitimate issue. Sometimes we'll go millions of years without seeing a fossil in a stripe, and then all of a sudden we see it again. Or we see something that looks like maybe it evolved from an ancestor, and there just are links missing. Also, we see punctuated equilibrium, where a lot of things explode out all at once. That's going to be hard to track in the fossil record. So the fossil record alone is not enough to establish any sort of evolutionary pattern. It's a piece of the evidence, piece of the case. Another type of evidence is homology or homologous structures. If you look inside these appendages, you'll see that they have a really similar bone structure. Over time, these organisms have used these structures differently, so they've become different. But notice the forelimb is highlighted in all four organisms. We've also got a similar setup here, 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 and here. One upper arm bone. I'm saying arm, upper appendage bone. The quote-unquote fingers or toes, whatever you want to call it, have modified over time, and so they become different. This is called divergent evolution. You have a common ancestor, common structure, you use it differently, so the structure then becomes different. So issues with homology, we see things called vestigial structures. And these are essentially evolutionary leftovers. Whales have legs. Snakes have legs that the bones tuck up between the rib cages. Humans have tails. Here's an example of that. You see a human tail. That's called a vestigial tail. It's not functional in any way, but it's there. So from an evolutionary standpoint, as we develop traits, we use them. As we need to develop traits, we do. When we no longer need them, gradually they're going to go away. but not instantaneously. Humans haven't needed a tail for quite some time, and we still have humans born today. I've had students that have been born with vestigial tails, so they stick around. Yeah, so that's kind of what that slide talks about. So homology in embryos has been a controversial idea, and it's really frustrating to most scientists because it didn't need to be. The scientist that originally published this work fudged his results. He made the drawings look more similarly than they were. And it wasn't necessary. If we track using today's technology, if we take photographs, we don't Photoshop them, we take photographs of different organisms as they develop, they are really, really, really similar, like strikingly similar. Here you're seeing a chick embryo and a human embryo, and you can see the things that are pointed out in the picture, and these are really common in things like pigs, fish. Lots of different organisms follow a similar developmental pathway. And again, the logic behind that being evidence for evolution is a pig would grow to be a pig, a snake would grow to be a snake, a human would grow to be a human in independent ways unless they had a common ancestor. It doesn't make sense for them to have a common growth plan if they didn't have a common ancestor. They're not going to the same place, so why would they follow the same path most of the way? Yeah, so I left this slide in because I wanted to discuss the fact that this connects to our Hox genes that we talked about in genetics. So make sure if you don't remember what Hox genes are, you take a look back. Yeah, so I talked a little bit about divergent evolution, how things become different over time as you use them differently. Kind of the opposite of that is going to be convergent evolution. So that's where distantly related organisms are going to show similar adaptations. Here's kind of the classic example. Sugar gliders and flying squirrels are really similar organisms that don't share a common ancestor, a recent common ancestor. So they've developed these traits because they needed them. Over time, they gradually were able to glide from tree to tree. This is kind of another piece of evolution. We talked about this in class. Some of you guys pointed out the fact that we see marsupials in Australia that are unique to that area. We see species mixtures on islands. We see anytime we see islands, we see weird things. So here's an example of Drosophila, which is the fruit flies that we looked at in genetics, and you can see how they migrated from island to island in Hawaii, and how they changed. So now we've got 4 different species over about 5 million years. Remember that each of these, the demarcation is going to be when they can no longer breed to produce fertile offspring. I will pick up the last few slides in the next video.